was notified and had the host photographed, which clearly showed bloodied flesh and had somehow grown larger than the original host.
After putting a consecrated host under a microscope, the scientists found something that should have been impossible. These are Eucharistic miracles that left scientists speechless for centuries, and they're still happening today. If you want to explore more scientific miracles, hit that subscribe button before we begin. The start. It was the year 750 AD in Lanciano, Italy. Here, a Basilian monk was standing at the altar in doubt. As a priest, he was supposed to believe that during the Mass, bread and wine literally transformed into the body and the blood of Christ. However, in his heart, he questioned whether this could really be true. Interestingly enough, as he spoke the words of consecration, something amazing happened. According to tradition, the bread in his hand physically changed into flesh, and the wine transformed into what seemed like blood. The monk was standing there frozen, witnessing what seemed impossible. The flesh and blood remained visible, and they have been preserved in the Church of San Francesco in Lanciano ever since it happened. For over 1,200 years, these relics have existed as a religious faith. But in 1970, something changed. The Catholic Church decided that it was time to apply modern-day science to this ancient mystery to see what they could find. Then, Professor Odoardo Linoli was chosen for the task. He was the head of the Laboratory of Clinical Analysis and Pathological Anatomy at the Hospital Arezzo. Being a full professor of anatomy, histology, chemistry, and clinical microscopy, he wasn't some regular researcher. Rather, he was respected among scientists with decades of experience. And now he was about to examine something that shouldn't even exist.
The year 750 AD. During the consecration at the holy sacrifice of the Mass, a priest in Lanciano, Italy, who doubts the real presence of Jesus Christ in the Holy Eucharist, is stunned when the host transforms into human flesh and the wine transforms into human blood.
When Linoli extracted parts of the relics and analyzed them, he found something amazing. He confirmed that the flesh and blood were of human origin, and the flesh was, without a doubt, cardiac tissue, specifically from myocardium, which is the muscular wall of the heart. After months of examination, he concluded that the flesh was also a human heart muscle that had claws because of human blood, both blood types A and B. From there, he published his findings in March 1971 in the Italian Medical Journal. The reports documented something that was almost absurd. This wasn't just any tissue. The analysis revealed that an internal hollow organ record was recognizable through a transverse section, historically identified as a heart with a left ventricle appearing much thicker. They were looking at what happened to be a cross-section of an actual human being. However, here's what made it even stranger. This tissue was over 1,200 years old, without any preservation, and it should have decayed decades or centuries ago. But interestingly, it was still there, intact, identified as cardiac tissue. Modern science. It ended there, except that something remarkable happened in the 1990s. Eucharistic miracles started occurring again, and this time they happened in an era with even more advanced scientific capabilities. In 1992, in Buenos Aires, Argentina, a consecrated host was found in a candle holder at the back of a church. He was following proper protocol, and the priest placed it in water, where it should have dissolved. But they were shocked to find out 1 week later, after they opened the tabernacle, that the host had transformed into a red blood-like substance. The local bishop at the time was George Mario Bergoglio, who later became Pope Francis. He ordered the substance to be photographed and preserved. 4 years later, in 1996, another incident occurred similarly. A host had fallen into the ground and was placed in water. Days later, it too had transformed into what appeared to be blood tissue. The same bishop decided that this phenomenon required serious scientific investigation. The samples were then sent to laboratories in New York, where specialists examined them without knowing their actual origin. The scientists were only told they were analyzing tissue samples of unknown origin from Columbia University. Dr. Frederick was a renowned forensic pathologist and cardiologist. Reportedly, when he saw the tissue under the microscope, he made an amazing observation. He told the doctor who brought him the sample that if the white blood cells were present in the heart tissue, it meant that at the moment the sample was delivered, it was still pulsating, as if it were a living heart. When Dr. Frederick was finally told the origin of the sample, he reportedly fell silent for a few minutes while being visibly shaken. This evidence was so overwhelming that the doctor was unable to speak for 5 minutes. He said outright that this is naturally impossible if you say that it is true. The 2006 findings. After this experiment came 2006, and what would become one of the most thoroughly documented Eucharistic miracles in the history of mankind. This time it happened in Textila, Mexico, during a spiritual retreat at St. Martin of Tours parish, where a nun was distributing communion after she noticed something unusual. The host in her hand had begun to use a reddish substance. She immediately showed it to the priest, and the bishop was immediately called to examine it.
Ordered an investigation and invited Dr. Ricardo Gomez to head up a scientific study to verify the event.
This time, it was Bishop Alejo Zavala Castro who looked at the Theological Commission and ordered extensive scientific testing. The samples were analyzed by multiple forensic medical laboratories across Mexico, Guatemala, Bolivia, and even the United States. The scientists conducting the test were not told where the sample came from, and they didn't know that even their testing was being tested. Dr. Ricardo, who was an atheist neuropsychologist, was brought into the equation this time for investigation, but he found that not only did it change the world of Eucharistic miracles, but also his life. The analysis revealed that there was a thin filament of tissue in the center of the host from where the blood was coming out. The tissue was alive with the living blood. However, the tissue died shortly after being disconnected from a body, and died shortly after being disconnected from the body, and the presence of living tissue and blood after 3 months was scientifically unexplainable. The test revealed even more bizarre details. The same tissue exuded fresh blood from inside to outside of the host. Blood on the host's exterior was produced in 2006, while blood on the inside of the host had remained fresh as of 2010. The pressure of natural flow from inside to outside was something that was unscientifically possible. Think about what this means for a second. Blood doesn't behave this way. Once the tissue is separated from the circulatory system, it dies almost immediately. Blood soaks, and cells start to break down. Here was a tissue that had been producing fresh blood years after its initial events, as if it were connected to the living body. And before we talk about what happened next in Poland, please make sure to subscribe to the channel and ring the bell icon if you like videos where we uncover scientific miracles. Miracle in Poland.
Who was about to receive the Eucharist pointed it out to him. According to church protocol, the host was placed in water and stored in a safe place. The reason the consecrated host is placed in water is that Jesus remains present in the Eucharist under the appearance of bread until the appearance of bread has been dissolved in the water.
Finally, in 2008, another incident occurred, and this time it happened in Poland during Mass at St. Anthony's Church. A consecrated host fell to the ground. Following protocol, it was placed in water in a silver vessel and then stored in the safe. Just 1 week later, when the sacristan opened the safe, she noticed something strange. The host hadn't dissolved. Instead, a red stain had appeared on it. This sample was sent to the Department of Medical Pathomorphology at the Medical University of Białystok for analysis to Professor Maria Sobaniak and Stanislav Sulkowski. They examined it, and the results indicated the presence of human heart tissue with specific changes. The tissue fragments that could be observed under the microscope didn't belong to a human heart, and looked as if the sample had been taken from the heart of a living person who was in pain. However, there was something even more extraordinary about this sample compared to anything we've seen in the past. When scientists examined it on the microscopic level, they discovered something that should even be possible in both Textia, Mexico, and here in Poland. Miraculous host and heart tissue were intertwined on a microscopic level. The doctor in Poland, who was one of the examining experts, stated that in this miracle, the remaining host was tightly interconnected with the fibers of a human tissue, penetrating each other inseparably as if bread were transformed into flesh. Even the NASA scientists, who have at their disposal the most modern analytical techniques, would not have been able to artificially recreate such a thing. Just 5 years later, in 2013, something similar happened in Poland, where a consecrated host fell during the Mass after being stored in water. A crescent-shaped portion turned red again. It was sent for scientific analysis, and again, human heart tissue was identified. The pattern discovery. Dr. Franco Serafini, who was an Italian cardiologist, became fascinated by these cases. He spent over 8 years traveling the world examining Eucharistic miracles firsthand, meeting witnesses, and even reviewing original scientific documentations. According to his research, he wrote a book called A Cardiologist Examining Jesus. Serafini examined 5 Eucharistic miracles closely and found 4 common features that appeared 5 times out of 5. The presence of heart tissue, specifically myocardial tissue showing signs of suffering. Blood was also present in all cases, and the most common one was AB blood type, the same blood type that was found in the Shroud of Turin. DNA was also present, but it behaved in a very strange way, so they started calling him the elusive DNA. In several cases, there was evidence of trauma from the presence of thrombi, indicating repeated lack of oxygen. Unlike regular human DNA, the DNA in Eucharistic miracles is not fully legible when scientists try to amplify it with modern human-specific primers. It resisted analysis, so those were some of the things that were present, the opposing side. Even after so much evidence, not everyone accepted these findings and believed them. Dr. Kelly Kearse, a Catholic scientist and immunologist who trained at Johns Hopkins Hospital and worked as a principal investigator at the National Institute of Health, has raised some important concerns about this methodology that was used in some of the investigations. In papers published in Forensic Science, Generals Kearse called for more accurate research methods and standardized protocol for investigating these miracles, in order that in multiple cases, hosts were found in improper locations and forensic, placed in water according to the Church's norms for disposal. This environment could potentially allow bacterial and fungal contamination. One common explanation also suggested that the bleeding that happened suggested for bleeding hosts is Sariata marcescens, which is a bacteria that produces a red pigment that is called prodigiocin. Research has also shown that when non-consecrated wafers are inoculated with this bacterium, the reddish region starts to appear within 24 to 48 hours, which looks like fresh blood. However, the scientists that were involved in these cases specifically tested for this bacteria, and a lot of other ones too, ruling them through the doctor's staining and spectroscopy, which are two well-established techniques that easily distinguish bacterial growth from genuine blood. Whether we believe these miracles are true or not is up to us. However, there is definitely something up with these miracles happening again and again and again. What do you think about them? Tell us in the comments below. And if you enjoyed the video, please make sure to subscribe to the channel and ring the bell icon for more videos like these.